Today's laboratory topic will be about selective and differential media in microbiology. A differential medium is a medium that contains ingredients that allow us to visually distinguish microorganisms that perform or maybe do not perform a particular biochemical process. So we are looking for a process of differentiation that we can achieve in that medium by adding some ingredients that will facilitate, for example, distinguish between organisms that are growing there and that maybe can ferment a sugar or cannot ferment a sugar. And depending on those results, the microorganisms may produce colonies with different colors, or they may change the color of the medium. That will be the process of differentiation there. Now, when we talk about selective media, a selective medium contains selective agents, or one agent or more than one, that inhibit the growth of unwanted microorganisms while allowing the growth of those that we are trying to isolate. So another important aspect of microbiological media that we're going to discuss in this laboratory is the concept of complex versus defined medium. A defined medium is a medium in which all the molecules included in the medium are known. Now, if we are talking about a complex medium, here this medium includes at least one complex ingredient, and for this reason, the exact chemical constitution is not known. That complex ingredient could be extracts of plant or animal tissues, enzymatically digested animal proteins, like for example, peptone or tryptone. That would be an example of these complex ingredients. And to make this more clear, especially for the defined medium, for example, there is a medium, the name of this medium is M9 medium. It could be a broth. And if we look at the list of ingredients in this medium, you will see that, for that medium, you have glucose, and then a list of salts that are included in that medium. And then the pH is adjusted to 7, around 7. Here, each ingredient, we know that molecule and the chemical composition, the exact chemical composition of that medium. Now, in the complex, again, there is always at least one ingredient that is complex. And all the microbiological medium that we're going to discuss today, they are example of this complex medium. Let's start with this one, blood agar. Blood agar is considered a differential and complex medium. It is complex because the following ingredients, this one, this one, this one, and this one, are examples of complex ingredients. In each of them, we don't know exactly the composition of all the molecules that are part of that ingredient. So for that very reason, we consider this medium very complex medium. Now, in terms of being selective or differential, this one in particular is only differential. And it's considered differential because with this medium, you can differentiate the microorganisms that are growing in this medium based on the ability to produce alpha hemolysis, beta hemolysis, or gamma hemolysis. Before I go into explaining each of these kind of hemolysis, let's finish looking at all the ingredients in this medium. For example, it has sodium chloride to give the proper osmolarity to the medium. It's an isosmolar medium. It has agar, and remember that the function of agar in microbiological medium is always a solidifying agent. Is used to create that semi-solid medium, right? That we have there. It's not a source of carbon or energy. It's nothing like that. The main purpose of adding agar to the medium is to provide an ingredient that will help solidify that medium. The fibrinated blood in this particular medium is what allows this medium to be differential and allow us to differentiate the microorganisms growing there into these three different categories of hemolysis. And then, of course, the medium also has water. So what are these different hemolysis? Let's go one by one. And we're going to start with, let's start with alpha right here. Alpha hemolytic species are microorganisms that when you grow them in the presence of this medium that contains red blood cells, they cause oxidation of iron in hemoglobin molecules that are within the red blood cells. And that gives a greenish color on blood agar. And by that I mean, the agar under and around the bacterial colonies is dark and greenish. It's also called, by the way, green hemolysis because of the color change in the agar. And another name that is used for alpha hemolysis is incomplete hemolysis or partial hemolysis. So the alpha hemolysis is caused by hydrogen peroxide produced by the microorganism. And this is what oxidizes hemoglobin, producing the green oxidized derivative that is called methemoglobin. Methemoglobin. So this is the alpha hemolysis. Let's now talk about the beta. This one right here, beta hemolysis. It's also called, in this case, complete hemolysis, and is the complete lysis of red blood cells in the media. And as a result of that complete lysis, is going to show a clear, transparent halo around and under the colonies that are producing that beta hemolysis. This complete lysis is usually caused by exotoxins produced by bacteria. That is the case, for example, of streptolysine, an exotoxin produced by Streptococcus pyogenes, group A, Streptococcus, group A, Streptococcus. This exotoxin interacts with cholesterol in the membrane of eukaryotic cells, not only red blood cells, like in this particular medium, but in real life, you know, when the bacteria is producing this exotoxin, that exotoxin is targeting red blood cells, white blood cells, different type of cells. And it is thought to play an important role on not allowing the host immune system to clear the infection rapidly. Examples of bacteria that do this beta hemolysis, well, I mentioned here the group A Streptococcus, but for example, Clostridium perfringens that produces gas gangrene is also beta hemolytic. In the case of the alpha, I forgot to mention examples of microorganisms that do alpha. Alpha hemolysis, for example, Streptococcus pneumoniae is an example of a bacteria that produces alpha hemolysis. Let me think about another one. Streptococcus viridans, that's another bacteria that produces alpha hemolysis. And then the final example of hemolysis that we can differentiate when we grow microorganisms in this blood agar medium is the gamma. And basically, gamma is a group of microorganisms that they produce no changes in the medium. They don't oxidize the hemoglobin. They don't lyse the red blood cells. So they basically produce no change in that particular medium. Let's now look at the next medium that we are discussing today. It's called mannitol salt agar, or MSA. If you look at this medium, here we are in the presence of a medium that is selective, differential, and complex. Why is complex? Well, you have here beef extract is the main ingredient that is making this medium complex. And then it is selective and differential. So first, let's look for the selective agent or agents that are making this medium selective to grow only a particular group of bacteria. That selective agent is this one here, sodium chloride. If you remember, sodium chloride in the previous medium, in the blood agar, was at 0.5% to create that isosmolar medium. In the case of mannitol salt agar, we have 7.5, a lot of salt. So this is making this medium, get a high osmolarity. It's going to make this environment hyperosmolar. And you could imagine that if the medium is hyperosmolar, then only microorganisms that can withstand this harsh condition will be able to grow there. And is generally used for the selection of halo-tolerant bacteria. Now, the medium also has the mannitol, and this particular ingredient is going to be the ingredient, one of the ingredients that is going to be important in the differential quality of this medium. Mannitol is a sugar, and some of these halo-tolerant bacteria that will be growing there, some of them can use the D-mannitol and ferment that, and as a result of the fermentation, they produce acid. And if the bacteria that is growing there is able to ferment that D-mannitol and produce acid, this medium that is initially red will change color because of the presence of a pH indicator. pH indicator that changes the color of the medium, or also it could be, color of the colonies, depending on the production of not of this acid due to fermentation. When the medium becomes acidic because of the fermentation, then the medium will change color from red to yellow, OPH. And if the bacterium growing there is actually producing something that is alkaline and the pH start to increase, then now the medium will change from red to pink at pH higher than 8.5. 8.4. Once again, you see agar here, and once again, the only function of agar there is solidifying agent. Okay. Now this is the showing a fermentation pathway that the microorganisms that are using mannitol are following, and that at the end produces organic acids that will lower the pH. And again, some of these halo-tolerant bacteria that are growing here, some of them are able to ferment the mannitol, others are not. And then based on that, we can differentiate them. And this is an example of what may happen there. The top section of the medium shows the medium that has not been inoculated, and you see the color of the medium is red. But if all of a sudden you inoculate here a bacterium that is halo-tolerant, that can grow there in that high salt concentration, and that at the same time is able to ferment mannitol, you see how the color of the medium changes to bright yellow. And this is something that, for example, Staphylococcus aureus can do. However, if you inoculate other species of bacteria that is halo-tolerant, can grow there, but they cannot ferment the mannitol, you will see that the medium doesn't change, or like in the case of the Staphylococcus epidermidis, the medium gets a little bit alkaline as a result of the pH of the medium going up. So basically, to summarize here, if an organism can ferment mannitol, an acidic byproduct is formed, and that causes the phenol red in the agar to turn yellow. It is used for the selective isolation of presumptive pathogenic Staphylococcus species, like the case of Staphylococcus aureus. The Staphylococcus species that are positive in this medium are coagulase positive. And this is a test that I want you to investigate on your own, because you will have to use it in your virtual laboratory in the homework that you have to complete. Look for the coagulase test. Okay. Let's now go to the third Oh, by the way, this is a table that is summarizing what I just explained verbally in the previous slides about the mannitol salt agar medium. Let's now go to the third medium that we're discussing today. That is the case of eosine methylenbutene. That is also called Levin's formulation. Both are, two names for the same medium. This is a selective, differential, and complex medium. Why this is complex? Well, right here, that ingredient is the main reason why this medium is complex. Why the medium is selective? Well, these two ingredients are responsible for the selectivity. These two ingredients, they prevent or inhibit the growth of gram-positive microorganisms, making this medium selective for gram-negative bacteria. Those are the ones that can grow in this medium, the gram-negative bacteria. In addition to that, the medium contains lactose, and lactose becomes the differential ingredient because the lactose there is going to be used by the gram-negative bacteria that is growing there, can be used as a substrate to ferment. And as a result of that fermentation, acid will be produced, and the production of acid will change the pH of the medium, change in pH that will allow the colonies that are growing there to change their color. Once again, the medium also has agar, and the only function there is solidifying agent. Now, the EMB, again, it's differential because it can distinguish between gram-negative organisms that ferment lactose and those that do not ferment lactose. The rapid and vigorous lactose fermentation produces a lot of acid. And this production of acids, high quantity of acids lower the pH. And this encourages dye absorption by the colonies that are growing there, which are now of a purple black color. That's in the case of bacteria that are producing a lot of acid as a result of fermentation of the lactose there. So additionally, on EMB, if Escherichia coli is growing there, a fecal coliform, it will give a distinctive metallic green sheen that you can see there, metallic green sheen, that is due to the metachromatic properties of the dyes. Now, some gram-negative bacteria that grow in this medium, they may produce acid, but just a little bit of acid, not a lot. And as a result of that, they will show, this pink mucoid colonies. Then you will have those gram-negative that are non-fermenters, like cannot ferment lactose, and that instead of producing acid and decreasing the pH, they may actually increase the pH by deamination of proteins. And this ensures that the dye will not be absorbed, and the colonies in this case will be colorless. And once again, if the inoculant that you are inoculating there doesn't show any growth at all, then in that case, you will be in the presence of a gram-positive that is being inhibited by the selective agents that in this case are methylene blue and eosine white. This table, like in the other medium, is basically summarizing the potential results that you may get there, interpretation and the presumptive ID for the microorganism growing in this medium. Then let's now go to the last medium that we are discussing today. And this one is called the McConkey agar. Here we are in the presence of a selective differential and complex medium. Why is complex? Well, pancreatic digest of gelatin, pancreatic digest of casein, pectic digest of animal tissue, bile salts. Those are all ingredients that make this medium very complex. And why the medium is selective? What makes this medium selective is the presence of these two ingredients, bile salts and crystal violet. Similar to what happened in the EMB with the methylene blue and the eosine white, here, these two ingredients, bile salts and crystal violet, they prevent or inhibit the growth of gram-positive microorganisms, making this medium selective for gram-negative bacteria. So only the gram-negative bacteria can grow there, but now the gram-negative bacteria that may grow there in this medium, they can be differentiated based on the ability to ferment or not lactose. And the fermentation or not of lactose, the differentiation is going to be achieved thanks to the presence of a pH indicator that in this case is neutral red. This is not phenol red like in the MSA medium. In this case, this is a different pH indicator, neutral red, that produces different change in colors when acid is being produced. In this case, lac positive bacteria, that means those that can ferment the lactose, those gram-negative that can ferment the lactose. By utilizing the lactose available in the medium, the lac-positive bacteria, like for example, Escherichia coli or Klebsiella aerogenes, they will produce acid, which lowers the pH of the agar, and then this results in the cells and the colonies, right, that are being produced by this bacteria that is growing there, getting a pink color. Not only the colonies will show this pink color, the medium also will be turned pink as a result of this acid that is being produced. Also, the bile salts, they precipitate in the immediate neighborhood of the colony, causing the medium surrounding the colony to become hazy. And again, not only the colonies are turning pink, but also the medium around the colonies may also turn bright pink. Now, if the bacteria is not able to ferment the lactose, and here we are in the presence of lac-negative, gram-negative bacteria, then the organisms are unable to ferment lactose. They will form normal colored colonies, and you're not going to see this bright pink color, of course, because there is not acid being produced. Here you have an example of a lac-positive bacteria versus a lac-negative bacteria. And also a table summarizing what I just explained verbally about the interpretation and the presumptive idea of the microorganisms that will be growing or not in this medium. So the remaining part of this laboratory class is a hypothetical experiment. I have hypothetical results. And during our meeting, we're going to look at these hypothetical results. We're going to interpret these results.